Today we will discuss about the interrelation between ZEB-1 EMT and microRNA 200. So let us before going into the topic directly, let us understand what does each of these term means. ZEB-1 is the protein that helps in the suppression of epithelial characteristics. In other way it will promote mesenchymal characteristics of the cells. This EMT is the short form for epithelial to mesenchymal transition. So epithelial cells are the cells that remains at their position. However, mesenchymal characteristics, the cells when acquire their mesenchymal characteristics, they can move from one place to another place. And this transition from epithelial to mesenchymal is commonly termed as EMT or epithelial mesenchymal transition. On this last one is this microRNA 200. This number is important. This microRNA 200 has some kind of special functions, and this one of the special functions is that it will inhibit this ZEB-1 translation by disturbing the mRNA 3' untranslated region of this ZEB-1 mRNA. So that's why because of this, mRNA will not be able to the stability of the mRNA will get affected, and the translation of ZEB-1 will be reduced. So this is how this microRNA will impact this ZEB-1 formation. So now let us understand how they are interrelated. So whenever this microRNA 200 is expressed within the cells, what it will does is it will inhibit or it will destabilize the mRNA of ZEB-1 protein by disrupting the 3' UTR site of the ZEB-1 mRNA. And because of this, because of the unstable mRNA of the ZEB-1, the translation rate of this ZEB-1 will get reduced, and the function of ZEB-1 is to suppress epithelial genes will also get reduced, and cells which usually is showing more mesenchymal type of characteristic because of the expression of this ZEB-1 will get affected. The characteristic of those cells will get affected as the ZEB-1, because as ZEB-1 is not been translating at its normal rate, and because of this the suppression of epithelial genes is not proper, and the epithelial markers such as E-cadherin will starts to express in those kind of cells where microRNA 200 is expressed, and cells will again move from mesenchymal characteristics to epithelial characteristic. As we can see that this microRNA will prevent ZEB-1 formation, and the cells will again move towards mesenchymal characteristics as E-cadherin will starts to translate. Whereas if this microRNA is not there, this ZEB-1 will inhibit this E-cadherin translation, and E-cadherin is the marker for epithelial cells. So whenever the E-cadherin are expressed, cells are in the epithelial regions, and the cells will remain intact into their position, and cells will remain intact in their position because of the cell contact. As we can see there, the cell contacts remain intact in the case of epithelial cells, whereas in the case of mesenchymal cells, the phenotype of the cells changes, and this will impact the cell interactions. And another function of this microRNA is that it will also inhibit this TGF-beta 2 receptors, so that this TGF-beta will not be able to penetrate inside these cells. And this TGF-beta is also known to promote mesenchymal type of characteristics. So this microRNA will not only impact the ZEB-1, but it will also affect the TGF-beta 2 receptors or the mesenchymal type characteristics. Promoted by this TGF-beta will also be impacted in this case, as this microRNA. Now we know that this MicroRNA has two roles to prevent the mesochemical step characteristics. First, it will inhibit ZEB-1. That's thereby the suppression of epithelial genes will be reduced, and also this TGF-beta, which is also known as as a protein, which is also known as a cytokines to promote mesochemical type of characteristics. So its induction in the cells will also be reduced, as this microRNA will affect this TGF-beta 2 receptors. So this TGF-beta will not be able to enter inside the cells at the same rate. So thereby this mesochemical type of characteristic will be affected by this microRNA 200 at two different sites, or at two different factors. So I hope you got a basic idea about the ZEB-1, EMT and microRNA and how they are interrelated. If you need to understand more about this topic, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.